In this second part of the chapter on patterns of genetic inheritance, we are going to take a look at some disorders that are carried by chromosomes that are not involved in the determination of your sex, right? This type of chromosomes are called autosomes. Now, in disorders that are carried by autosomes, individuals that are expressing the disorders have to be homozygous recessive, right? For instance, in this pedigree here that shows you mom and dad and the offspring, we see that mom is actually expressing a disorder. Mom is recessive, right? Dad is not expressing the genetic disorder, is either homozygous dominant or heterozygous, right? Now, in that case, what we see in this pedigree is that this couple here had children, right? One, a girl here who is heterozygous, and another girl here who is heterozygous. Now, these two girls do not express the genetic disorder, which shows that most likely dad is probably homozygous dominant, right? But anyway, we do see that in going down the generations, we see that this individual and this individual are relatives. And these individuals who are relatives have children who are expressing the genetic disorder. Now, consanguinity, which is when there is relationship within the same family, right? For instance, between first cousins, chance to express a recessive genetic disorder is very high, okay? This is what we show here on this pedigree. But let us move on. For autosomal dominant disorders, only one allele is necessary to be present for that disorder to be expressed, so that the individuals who are expressing the disorders are either heterozygous or dominant, right? So if you have a mom who is heterozygous expressing a disorder, a dad who is heterozygous expressing a disorder, we see that the offspring are going to express the disorder, right? Some may not express the disorder only because mom is heterozygous, dad is heterozygous, right? Okay. Whenever the dominant allele is present, the disorder is necessarily expressed, regardless if the individual is homozygous or heterozygous. All right, so what are some examples of recessive disorders? One example is the Tay-Sachs disease. What is the Tay-Sachs disease? The Tay-Sachs disease, it's a neurodegenerative disease. That neurodegenerative disease occurs because there is absence of production of an enzyme that is actually responsible for breaking down some fatty acids that are found in the lysosomes. Now, the lysosomes are these organelles in your cells that contain digestive enzymes, right? The accumulation of those lipids in the lysosomes are causing in brain cells a dysfunctioning of nerve cells, right? In the brain, not just in the brain, but also in the spinal cord, right? In the central nervous system, let's put it that way, causes a neurodegenerative disease, right? And the onset of the disease is typically at age 20, something like that. But don't quote me on that. Another disorder that is associated with a recessive allele that is carried by an autosomal chromosome, is a cystic fibrosis. Cystic fibrosis, this is when chloride ions are abnormal, and chloride ions occur in the plasma membrane of epithelial cells, and these epithelial cells are typically positioned along the digestive tract, along the urinary tract, the reproductive tract, the respiratory tract. So, with the absence or the dysfunctioning of this chloride ion, you have a thickening of the mucus in the mucous membrane, which is actually going to cause frequent infections in individuals with cystic fibrosis. These infections can occur in the respiratory tract, they can occur in the digestive tract, they can occur in reproductive tract, and so on. So many possibilities, right? This is a pleiotropic kind of condition, right? Another recessive disorder is phenylketonuria. Phenylketonuria, it's actually caused by the lack of an enzyme that is needed to metabolize an amino acid that is called phenylalanine, right? That phenylalanine is an amino acid that is found in proteins. So as consequence of that, individuals with phenylketonuria, they have to have a diet that is reduced in proteins. Now, because of how technology has evolved these days, it is possible to actually track the amount of phenylalanine in some proteins so that individuals are going to avoid some type of food. All right. Now, this phenylketonuria has an effect on the nervous system, the development of the nervous system, so that ladies who are pregnant and who have phenylketonuria, they have to have a low diet in protein so that their offspring is not affected, because the offspring can very well be normal without phenylketonuria, but if mom does not respect a strict diet, well, the accumulation of phenylalanine is actually going to affect the development of the fetus. All right? So that the fetus can have a complete normal genotype, but the phenotype can be considerably affected, as if they themselves had phenylketonuria, just because mom did not follow properly the the restrictions, the diet restrictions during pregnancy. Anyway, another one is sickle cell disease, which is a homozygous recessive affection where red blood cells, rather than being biconcave, they're going to have a sickle shape. So biconcave, so cells typically are like this, right? Red blood cells. Now, in the case of individual with sickle cell, the cells are going to kind of be shaped like this, which actually causes a problem when navigating blood vessels, and particularly the blood capillaries, right? These edges are actually hurting the walls of the blood vessels and causing all kind of inflammations and issues. It's pretty difficult situation. Anyway, let's move on. Okay. In the case of the Tay-Sachs disease, so here we have lysosomes that have the normal enzymes and are breaking down fatty acids, right? But in an individual with the Tay-Sachs disease, we have an accumulation of the lipids in here so that the lysosomes that are supposed to break down these lipids are not breaking them down, which is causing the neurons to dysfunction, okay? Causing a neurodegenerative disease. All right. Now, what are some autosomal dominant disorders? One example is the Marfan syndrome. Marfan syndrome, it's a condition in which elastic fibers are dysfunctional, right? Elastic fibers are part of the connective tissues in human, and one particular protein in elastic fibers is called fibrillin. So the fibrillin is not properly shaped, and that lack of integrity of fibrillin can cause multiple affection in the body, including dislocation of the lens of the eye, which is going to cause an individual to not properly see. Limbs are going to be unusually long, with long fingers. Some individuals are going to have caved in chest, and some individuals are going to have weak walls of the aorta, which can cause the aorta to burst, which is a major problem you can imagine, right? Another dominant disorder that we can mention is osteogenesis imperfecta. Osteogenesis imperfecta, this is when the bone development is not happening properly. This is possible because of a defect in the synthesis of collagen. Collagen being one of the proteins that are produced in the matrix that make up our bones, right? That defective collagen is going to cause the bones to be weak and bristle so that they're going to break down easily, right? They're going to break easily. Fractures are going to happen easily. And another disease that I would like to mention is the Huntington disease. The Huntington disease, it's a disease that is caused by a protein that is called huntingtin. That protein, huntingtin, it's actually carry too many amino acids that are called glutamine. Now, you have a number of glutamines in the protein huntingtin. However, in the defective huntingtin, they have just an excessive amount of glutamine. And the more glutamine they have in excess, the individuals, the more quickly the onset of the disease is happening in an individual, right? That onset, it's actually a progressive neurodegenerative disease, right? That occur in the central nervous system and particularly the brain. All right, so here in the case of the Marfan syndrome, we are going to have some connective tissues that are going to be affected. There is a particular protein that we call fibrillin. Fibrillin, all right? That protein is going to cause all kind of affection in the skeleton, the heart, the blood vessels, the eye, the lens of the eye that gets dislocated, the lungs, the skin, and you have really a bunch of consequences here that I'm not going to list, right? But for your own curiosity, you can take a look at that. So this right here, it's collagen. This is a collagen fiber. This is right here, elastic fiber, another elastic fiber, another elastic fibers, okay? This is some loose connective tissue that is shown here. All right. Okay, another genetic disorder here. We have cystic fibrosis with the chloride ion channels that do not function properly, so that the proper balance of chloride ion is not kept across the plasma membrane. As consequence of that, we have a thickening of the mucus, which causes individuals with cystic fibrosis to trap a lot of the debris that we breathe in, or that are in our digestive tract, causing the both the digestive tract and the respiratory tract to be prone to infections. And here we have the case of Huntington. So here in a normal brain, you can see how the brain cells are close to each other, while in the case of Huntington disease, how brain cells are really distant from each other, showing, a breakdown of this brain cells. All right. Now, these days, because of all that we know about patterns of genetic inheritance, a lot of parents these days are doing testing of embryos, of eggs, sperms, before actually fertilization occurs and after fertilization occurs, right? Pre-implantation genetic diagnosis, it's an approach that allows parent to see if an embryo they are considering for implantation, or a fertilized egg that is being considered for implantation, is having a genetic disorder or not, right? Now, obviously, one can argue that, this may pose some ethical issues, but that's another story that I'm not going to dive in, right? So there are a number of things that can be done, including following in vitro fertilization, right? After the zygote divides, some cells of the zygote can be obtained and analyze this zygote for some genetic disorders, right? I mean, this cell, this embryonic cell for genetic disorders, right? For to analyze this cell has to be grown after it is isolated from the embryo. It will have to be grown so that there is enough DNA for testing to be done. So typically, when that happens, parents are only going to keep embryos that will not express the genetic disorder, right? For implantation in the uterus. All right, so you are shown here a mass of cells with one cell that is isolated, and that cell is grown in a lab for analysis, so that if everything is healthy, then they're going to say, okay, well, now let's implant the embryo in the uterus. Another option, it's that actually the egg is going to be analyzed, sperm cells are going to be analyzed, then only the eggs and sperm cell that are normal are the one that are going to be obtained for fertilization, right? And after fertilization, the embryo is actually implanted in mom for in the uterus for the production of a baby. All right. Now, let's take a look at polygenic traits and the influence of the environment. What is a polygenic trait? Poly means several. Genic means genes, right? So you have a particular feature, such as the color of your skin. That color of your skin is not determined by one single gene. It's actually multiple genes, okay? Multiple genes that occur in different kind of alleles. You have dominant alleles, you have recessive alleles. So when it comes to things like color and height, every single dominant allele is going to account for some aspect of the phenotype. And a recessive allele is also going to account for a certain proportion of the phenotype, so that we are going to have not two phenotypes, but a myriad of phenotypes, right? Multiple phenotypes, so that we are going to have a continuous variation of the phenotypes in the population. That continuous variation is going to actually describe what we call a bell curve. A bell curve. Now, that bell curve shows that you're going to have a very limited number of individuals for a given skin tone, right? Who are going to have a very light skin. A very limited number of people are going to have very dark skin, while the vast majority is going to be really in between, right? And you see that actually the number of capital letters and lowercase letter plays a very important role in the variation. So if you consider, for instance, a population of Caucasians, right, in Tuscarawas County, or even in New Philadelphia, to be a little more specific, right? You take a population of Caucasians in New Philadelphia, they all live in the same area, they mingle together, but they don't have the same skin tone, even though they are all Caucasians, right? They don't all have the same skin tone. You're going to have some individuals who are going to be very light-skinned, other individuals who are going to be very dark-skinned, but you're going to have many that are going to be in between, and really there is no one single skin tone that is exactly the same. So anyway, so we have that polygenic trait, and that polygenic trait is also influenced by environmental factors. For instance, the amount of exposure to sun. Someone, for instance, who is building roads, spending a lot of time outdoor, is going to have a very different skin tone compared to a relative, a sibling, who actually spend their time in a closed office without windows, right? So they're going to have very different skin tones. All right. Now, in terms of polygenic traits, now we are going to go from polygenic traits to polygenic trait and multifactorial traits. So polygenic traits, many genes, and influence of the environment, for instance, your diet, right? We notice that these days, children tend to be much taller than their parents. Well, if you consider, for instance, parents who were born during the Great Depression, and you consider these parents and their children who were born way after the Great Depression, well, during the Great Depression, there was not much food, people were starving, and so those children who grew up starving, well, have a phenotype that is affected by the fact that they did not have much, even though they ended up having a good job later, right? Having a good education and a good job. But their children, now who have a better diet than the parents, are now so much healthier and so much bigger and taller that, because of diet, these individuals are kind of in a better spot in their lives. So when we consider height, it's also a bell curve, right? Very limited number of adults are extremely short, right? 5'0" or 5'1", okay? While very limited individuals are going to be extremely tall, 6'4", 6'5". Most individuals are going to be, in this area, 5'5", 5'6", 5'7", 5'8", 5'9", 5'10", and so on and so forth, right? So we actually represent the bell curve right here, right? With most individual in the population that are going to be within a close close side of the height of the peak. All right. Now, some threats are controlled by the environment. For instance, in this Himalayan rabbit, this Himalayan rabbit is actually expressing melanin only in parts of its body that are cold. So the gene for melanin production is expressed at lower temperature, right? You notice that the ears, the snoot, okay, the paws, okay, are going to be dark because these areas are where temperatures are low, right? As consequence of that, the gene is going to be expressed in this area. Pretty interesting, right? Okay. While in the main part of the body where temperature is at 37 degrees Celsius, well, there is no expression of melanin because high temperature shuts down the expression of melanin. All right. So other cases of genetic inheritance or patterns of genetic inheritance, we have the concept of incomplete dominance. So in an incomplete dominance, you have that the allele you receive from mom and the allele you receive from dad produce an intermediate phenotype, right? You consider an individual with curly hair and an individual with straight hair, right? The offspring is going to have wavy hair, okay? That's an example of incomplete dominance. And then we have the case of codominance. So one is not more expressed than the other, okay? In the case of codominance, we are going to have here the case of family hypercholesterolemia. In family hypercholesterolemia, in an individual who does not have hypercholesterolemia, in the surface of our cells, we do have low density lipoprotein cholesterol receptors. Low density lipoprotein cholesterol receptors. These LDL cholesterol receptors occur in two copies. One you receive from mom, one you receive from dad, right? So in a normal individual, we don't typically have a high level of LDL in our blood because the LDL cholesterol receptors are going to internalize that cholesterol. Now, if an individual receives a mutated copy, say H is for the receptor and lowercase h is for the mutated receptor, right? An individual who is heterozygous is capital H, lowercase h. that individual is expressing half the number of receptors. Why? Because, let me show you here. Why? Because this individual is producing H from one of the parents and not producing H from the other parent. So only half. And as consequence of that, you have a high level of LDL in the blood, but an individual with who is homozygous, right? So that individual would be lowercase h. That individual does not produce at all LDL cholesterol receptors, so that they're going to have, deposits like this, okay? That is a major problem. And this individual typically are not going to reach the age of adulthood, right? Because of the cluttering of blood vessels and eventually death at a very early age. All right. In the case of codominance, the two alleles are expressed at the same time when they are present, right? Say we do have an individual who is type blood A, and that individual is homozygous. Another individual is type blood B, that individual is also homozygous. Now, when these two individuals come together, say this is mom and this is dad, they come together, their offspring is necessarily going to be AB, right? Because mom is homozygous for A, dad is homozygous for B, right? The sperm cells and egg cells are going to be A and B, so that the individual is AB, right? On the surface of the red blood cells, we are going to have A and B in these individuals, right? Okay. So this is A, this is B on the surface of red blood cells. All right, this is the case of codominance. Now, what is interesting though, it's that in a case of ABO, blood type ABO, we are in a particularly odd kind of situation. Why do I say this? Because blood type, it's not determined by two alleles, but it's determined by more than two alleles. Okay? So the gene is going to exist in several allelic forms. Okay? So that you're going to have multiple kind of phenotypes and multiple kind of genotypes, right? So how is this specifically happening? It's what we are going to take a look at in the next few slides. Okay. Say we have an individual who is A and another individual who is B. But the individual who is A is not homozygous A. The individual who is B is not homozygous B. Now, if you cross these two individuals, you are going to have some interesting blood types in the offspring, right? How so? You are going to have a possibility of A and B, a possibility of A and O, so A, B, A, O. You're going to have a possibility of O B. So O, B. And a possibility for O, 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 O. Right? So that you have all these different genotypes and the blood types that go with them. AB would be blood type B. BO would be B. AO would be A. And OO would be O. Now, what does this mean really rigorously? Rigorously, a parent who is A heterozygous is going to have capital IA and lowercase I. These are three different alleles. A parent who is going to be B heterozygous is going to have capital I, capital B, and lowercase I. These are three different alleles, right? So now, if you have these two parents that you cross together, dad and mom, what happens is that mom is going to give some gametes like this, some eggs like this, and some eggs like this. Capital I, capital A, and lowercase I. Dad is going to give capital I, capital B, and lowercase I. In terms of offspring, we're going to have capital I, capital B, and capital I, capital B. This is an individual who is AB. Here, we're going to have capital I, capital B, lowercase I. This individual is going to be B. Capital I, capital A, capital lowercase I, right? This individual is going to be A, while this individual lowercase I is going to be O, right? That's what we saw earlier, okay? So the phenotypic ratio is going to be 1, 1, 1, 1. 1 AB, 1 blood type B, 1 blood type A, and 1 blood type O, right? All right, so now we are going to address the sex-linked inheritance. In sex-linked inheritance, now we're going to take a look at the chromosomes that determine our sex. In this case, it's going to be X and Y, right? A female in a human is XX. A male is XY, right? There are some genes that are carried by X chromosome, or the genes are carried by the Y chromosomes. Now, let's remember that in a humankind, females have rigorously homologous chromosomes when it comes to sex chromosomes, XX. But males do not have rigorously homologous chromosomes because the genes that the X chromosome carry, the Y chromosomes do not carry them, and vice versa, right? So that these two chromosomes, while they are homologous, they are only homologous by their function, never by their content or their size, okay? So that some genes are going to be systematically expressed in some individuals while never expressed in other individuals. Well, let me not say never. But what I mean is that because X is not equal Y, anything that is on X in a male is going to be expressed. Let me repeat that. Anything that is on an X in a male is going to be expressed. But in a female, it depends, right? It depends because we're going to ask the following. Is that disorder recessive or dominant? That's the question, right? That's one. Two, it's, well, what is the genotype of mom. Is mom homozygous recessive, or mom homozygous dominant, or mom heterozygous? Right? Now, for dad, whatever is carried by X is what is going to be expressed, whether it is dominant or recessive, right? Because there is no equivalent in Y to counteract what is happening with X, right? Okay, so the Y-linked inheritance are always associated with males, never with females. The X-linked inheritance can affect both males and females, right? Now, most sex-linked traits are going to be carried by the X chromosome. All right, let's take a look at color blindness. Say we have a mom who has a normal vision, right? She's heterozygous, right? X capital B lowercase b, carried by the X chromosome. Dad has a normal vision, right? Capital B and Y, okay? So what are the possibilities in the offsprings? The possibilities in the offsprings, when you cross mom and dad here, what we do see, it's that in the offspring, we're going to have individuals who are going to have a normal vision. Here, female, homozygous, dominant. Normal vision here, female, heterozygous. In terms of male, normal vision here, and colorblind here, right? So if mom is heterozygous, chance that boys are going to be affected with a recessive disorder is 50%, because, well, there is actually, mom can give, where is that? Okay, mom can give this capital B or this lowercase b, right? For a son to be colorblind, I'm sorry, you need lowercase b to be crossed with a Y, right? And this will happen in 50% of the cases, right? So that's why 50% of boys have a chance to have to be colorblind, while 50% of chance in boys exists to have normal vision. All right. Okay, so this is a pedigree that shows color blindness, with a father with color blindness, a mom, grandfather with color blindness, and grandmother who is not color blind. When we look at the offspring, we see that the daughter here is not color blind, normal vision. The boy here has a normal vision, right? Because the grandma here is homozygous dominant, right? However, when this boy here, right, has a child with a lady with color blind, right, all the boys, regardless, are going to be colorblind, because mom can only give some gametes that are, that's not what I wanted to do, that are XB for one of the gametes, and another gamete that is X lowercase b, right? So for all the boys, they're going to be necessarily YXB, YXB. So all the boys are colorblind, right? No question about it. Okay. All right. So some color blindness. Okay. Well, some disorder. We have color blindness. Okay. That color blindness often occur in the red and green color blindness. Another example of X-linked disorder is going to be the Duchenne muscular dystrophy, which is the wasting disease, right? That wasting disease, it's basically the loss of ability to use one's legs because the muscles are actually, there is no tone to muscles. The Fragile X syndrome, which is actually causing mental impairment, and hemophilia, which is a condition by which some clotting factors are absent, right? And causing an individual to bleed. Even with a paper cut, the individual are going to bleed extensively. So remember that X-linked disorders are often found more in males than in females, right? Because the recessive alleles are always expressed in males. I want you to remember that. All right, so here we have an individual with a normal muscle tissue, right? Skeletal muscle tissue. And you can see how the muscle fibers are close to each other. They are compact against each other. While in the wasting disease, in this little boy, you can see how the muscle fibers are spaced out, right? And causing actually the muscles to not have the proper tonicity. All right, that's about it. Thank you for your attention. Bye now.